That's an important disclaimer. I'm an environmental engineer, kind of dabbling in bioinformatics and microbiome analysis. The reason I'm interested in it is that I'm looking at characterizing fungi in activated sludge wastewater treatment systems, because fungi are typically understudied in those systems, and they can provide some unique metabolisms to help degrade more recalcitrant compounds like pharmaceuticals and personal care products that tend to pass through and get into our waterways. So that's my motivation for being interested in microbiome research. But I'm not going to talk about that today. I'm going to introduce you to Chime 2, which unfortunately I haven't had a chance to use in much depth yet. So you might be asking yourself why I'm talking to you about it as an environmental engineer who hasn't used this version of the program much. And the reason is that last semester I won the poster contest at the networking meeting. And so the center was kind enough to support my attendance at a workshop, and I chose the Chime 2 workshop knowing that it was coming out and was going to be replaced. So I'm going to go through kind of high-level changes in Chime 2 versus Chime 1, kind of introduce some of the new features, and then maybe we can have a discussion about your experiences with it or with other alternatives to Chime.
So some of us won't even know what Chime 1 is.
Yeah, so I'll start with a little bit of background about that. So first of all, how many of you have heard of Chime? Okay, how many of you use Chime in your research? A couple. And has anyone used Chime 2 at all? Okay, so a couple of people. So Chime stands for Quantitative Insights in Microbial Ecology. It's pronounced Chime, but spelled Q-I-I-M-E. It's an open source software pipeline designed for processing, specifically for processing marker gene survey data. So if you're doing metagenomics or metatranscriptomics, Chime is probably not the tool for you at this point in time. But they're looking to kind of expand its uses, and I'll talk about that more later. But we can use Chime in marker gene surveys from raw sequence data through interpretation. It wraps other software packages, so Chime itself doesn't do a lot of the bioinformatics analysis, but wraps common software packages that you're probably familiar with so that you can do all of your analysis in a single environment. So you use the Chime interface to interact with other software packages. It was initially released in 2010. It was spearheaded by Greg Capareso, who also led the workshop that I went to. So it was a nice experience to kind of see and interact with the person who helped to create this. It's been cited about 10,000 times, so it's one of the more popular programs for this type of data analysis. But there are other alternatives, like Mother is the major alternative that a lot of people like. I haven't used Mother, and this is not to say that Chime is better than Mother. I'm only presenting on Chime as one option for you. So I don't have a strong opinion. Maybe we could talk about that also. So first of all, most of you are probably familiar with this, but since we're dealing specifically with marker gene surveys, when we use Chime, I just wanted to go over a few basics here so that we're all on the same page. So marker genes or gene fragments are common to all members of a group of interest and can be used to characterize phylogenetic or functional diversity based on sequence variability. So typically, these marker genes are flanked by conserved regions so that we can design universal primers and amplify this gene from all of the species of interest in our environmental sample. So typically, we're dealing with environment. Common examples of marker genes are the 16S for bacteria and archaea, and ITS or 18S for fungi. I personally have targeted the ITS region. So this is a schematic of the ITS region, just showing we have two choices for variable regions here, and they're flanked by these conserved regions in the ribosomal RNA gene. So a typical workflow for a marker gene survey, we'll collect an environmental sample, a soil or stool, in my case, activated sludge from a wastewater treatment plant. We'll extract the DNA to get environmental genomic DNA, and then perform PCR to target our marker gene of interest. This is where marker gene surveys are different from like a metagenomic approach. We perform this PCR step, and we get marker gene amplicons, a pool of these for each of our samples. Then we go into library preparation. There's a lot of complex details here, so I'm gonna skip over most of that, but one of the main ideas is that we can tag or index all the amplicons from each sample so that we know where they came from, and then we can pool them all together, sequence them, and we end up with our raw data that goes into Chime. So our raw data is typically millions or tens of millions of sequences, all indexed so that we know where they came from. So if we have multiplex, we need to demultiplex so we can sort those reads back into the original samples. We need to merge paired ends if you did paired end sequencing. And we need to quality filter to remove unusually short or long reads or reads with poor base calls. We end up with our per sample quality filter data. Typically, we'll cluster that at something like a 97% similarity. So we take all of our reads and we compare them. Those that are 97% similar, we group together into what's typically called an OTU or operational taxonomic unit. And this kind of represents a hypothetical species in our sample. Then based on our OTU table, we can assign taxonomy to those OTUs, so we know which organisms they represent, and we can calculate different diversity measures, alpha diversity, beta diversity, generate our figures and numerical measures. So all that can be done within Chime. If you need to do further analysis, like pie crust or something, you can typically export the data from Chime workflow and go into something like pie crust. So this is pretty important. Chime 2 is now succeeded Chime 1. So Chime 1 is no longer supported as of January 2018. So if you're using Chime and you're still using Chime 1, it's highly suggested that you move to Chime 2 so that you can get full support with problems as they arise. Some of the high-level features of Chime 2 are that you're going to get the latest microbiome bioinformatics methods. The developers are focusing all their efforts on Chime 2 now. So anything that's new that's coming out is going to be incorporated into Chime 2, but not into Chime 1. It's pretty accessible through the online documentation. Chime 1 was as well. They have pretty solid tutorials and documentation on the website, and I'll link you to some of those resources at the end. There's a pretty active community of scientists and software developers and bioinformaticians who are active on the Chime 2 forum. So if you go to the forum and post a question, you'll usually get a response within a couple hours to a day. Some of the lower level features are that in Chime 2, they're introducing multiple user interfaces. So those of us who have used Chime 1, we're used to using the command line interface, which I'm comfortable with and I like, but they're trying to make this more accessible to other users. So there's also a graphical user interface for Chime 2 that's in a prototype phase of development. You can download it and use it, but it's not going to be fully featured for a little while yet. And then there's something else called an API interface, which I'm least familiar with. It's targeted toward data scientists who do a lot of Python coding, so you can also interact with Chime 2 through this API interface. One of the neatest features in Chime 2, I think, is this decentralized provenance tracking, and I'll talk a bit more about that. And then the plugin architecture is also new to Chime 2, so I'll go into a little more detail about what that means as well. So here are some visual representations of those different interfaces. So in the top left is the command line interface, which most of us are probably familiar with. This is an example of the GUI, which again is in a prototype phase, and the artifact API, which I can't comment on too much, but this gives you a sense of what these different interfaces look like. And then the plugin architecture. So Chime itself doesn't do any bioinformatics analysis. It only wraps other software packages. So all bioinformatics analysis is defined in plugins. There are Python 3 method annotations. I'm not a programmer, so I can't go into much more detail about that. But the utility of this is that any developer can create a plugin. So if you have a method that you're using in your lab that you think would be useful to the general community, you can contact the Chime 2 developers, and they seem very keen on working with people to incorporate new methods into the platform. And this is a list of, this is my Chime installation, and there are some default plugins that come installed. And you can see some things you probably recognize, like Cutadapt, Data2, Blur, vSearch, and then some of these are more specific to Chime, like diversity feature table. And I'll show you how these are used when you're entering commands in a little while. So another thing that is new to Chime 2 is this concept of artifacts and visualizations. So in Chime 2, they've introduced two new file types. One is a QZA and one is a QZV. So QZA is a Chime artifact, and it basically contains data that's to be used for further analysis. The QZV is a visualization, so that's the output that you want to visualize. I was kind of leery about specific file formats when I first heard about this, because it seems to kind of constrain your work a little bit. But as it turns out, these are really just zip files that contain a bunch of additional information in addition to your data. So your data can be found in here. This is the extracted file. The data can be found in here in the data directory. In this case, phylogenetic tree. And the rest of the information in the zip file is human-readable, Chime-specific metadata, mostly related to the data provenance. So I'll show you a couple of examples of that. So data provenance is basically what that means is just keeping track of all the source data that went into your analysis and also all of the operations and actions that you performed to get to your final output. So even if you put a lot of effort into keeping good records, sometimes 4 months down the road when you're writing your paper, it's difficult to remember every detail of what you did to get to that figure that you're publishing, for instance. But Chime 2 helps in tracking all of that. So I showed you this one file was your data, and all of this is related to provenance. So another way to look at this is this visual diagram. And on the left is this object that you've created, right? It's either a QZA or a QZV file. So within the zip file, you have your data, as I showed you. You have your metadata related to additional information about your samples. You have version information, and then you have the data provenance. So within the provenance for this top level object, you have information about the artifacts that went into, artifacts meaning other data objects, other QZA files, that went into creating this object, and all the actions that were performed as well. And this is kind of a nested structure. So then for each of these, you also have provenance data. So it tracks it from the very beginning all the way up to whatever it is that you produced here. And each of these objects has a UUID, or a universal unique identifier. It's a really long hexadecimal random number that uniquely identifies all the objects and actually all the actions as well. And as I'll show you, there's actually a UUID for all the features, all the OTUs that you generate. So in addition to that, another big addition to Chime 2 are some web tools for collaborating. So you might want to share some output that you have with your supervisor, or you have a colleague or a collaborator that you want to share some visual output with. And you can do that through this website, view.chime2.org. It looks like this. You can either drag a file from your desktop to this space, or you can enter the web address for a Dropbox file, a file in Dropbox, or any file from the web, actually, if it's on a server somewhere and you have a web address for it. You can just send your colleague that web address, and they can enter it into this website, and it will actually download the data to their local machine and then do the processing and display it to their browser. So the data is never uploaded to the Chime server. And so that's a way of ensuring that your data is secure, that it's, it's not being placed on machines across the country. It's only on your local machine. And most of these visualizations are interactive. So I'll show you a couple of examples of how you can interact with the QZV files. So I'm going to show you some example commands and play around with some of the output. This is mostly based on this moving pictures of the human microbiome tutorial that is available on the Chime website. And I'll link you to those tutorials at the end. I just wanted to show you this and give you a little background so that as we're going through the output, it makes more sense. This project that the tutorial is based on had 2 human subjects that were sampled over a period of 6 to 18 months. And they sampled the tongue, the left and right palm, and the gut via feces. So before we get into entering commands, we of course have to have our metadata set up. There are some differences in Chime 2 versus Chime 1 with metadata. So it used to be referred to as the mapping file, but now they're just calling it the metadata file. It used to be very specific in that you had to have these 2 columns, barcode sequence and linker primer sequence, and you had to have a description as the last column. Those are no longer requirements. This barcode sequence and linker primer sequence is kind of specific to the way that you prepare your library. So if you're using the Earth Microbiome protocol, you'll likely have these columns, but if you're doing like a dual indexing approach and using the facility on campus, you're not going to need these columns. So you used to have them and leave them empty, which didn't make sense. But now the only requirements are that you have a column containing your sample IDs, and that each of your sample IDs are unique. You can't have redundant sample IDs. And that each of your other columns has its own header or heading. And another addition is that the second row here, so the type of data. And here you specify whether your metadata is categorical or numeric. If you don't specify, Chime will actually try to figure out whether it's categorical or numeric. But to me, it makes sense to just take that extra step to fill it in to avoid any problems. In Chime 1, there was a way to validate your mapping file, so you can make sure there were no errors before you went into your analysis. And they've developed a Google Sheets plugin so that you can do that with new metadata format as well. I don't personally use Google Sheets, so luckily there's another option here. Anytime you use metadata in Chime, it actually validates that file just before it's used in the calculation. So you can do this for peace of mind using the Qiime plugin, or you can just proceed and you'll be warned if there are errors in your metadata, before the analysis takes place. So this is just an example of that Qiime plugin. So if you have your metadata in Google Sheets and you install and run the plugin, you'll look for errors and then it'll announce the error, what type of error it is, so that you can fix it. So here it's saying that we have duplicate sample IDs in row 2 and 21, but it didn't show row 21. So you just need to change those sample IDs so that they were all unique. So once we have our metadata, then we can start to work with commands. Can you guys see that in the back? Probably very small. Sorry about that. So the first thing you have to do is import the data. The way you enter commands in Chime 2 is you first call Chime, and then the second entry is the name of the plugin, so the tools plugin in this case, and then the action within that plugin. So this action is import. So we're going to import our data. You want to specify the type. So in this case, using this tutorial, this is Earth Microbiome Project type data. So they have a type for that, EMP single-end sequences. And then you specify the input path and the output path. So if you notice, the output path is a .qza file. So we're basically taking our data, our raw data, and converting it into a QZA file so we can continue to use it with Chime 2. Typically, as I mentioned before, we need to demultiplex. So there's a separate plugin called demux for demultiplexing your data. Again, you need to specify the type, in this case, EMP single, but there are other types for other types of data. It's a different method to prepare your library for sequences. And here we're just inputting the QZA file that we generated in the import data stuff. So we're also specifying here some barcode mapping information. I don't want to go into a lot of detail. These two steps are going to be widely different depending on how you sequence. You probably need to go to the website to get some help on how to do this for your specific data. But the output here is the demultiplex data, another QZA file, demux.qza. And then we have the option to summarize our demultiplex data. So using the same plugin, demux, we can use a separate action called summarize. And we can input our QZA file from the demultiplex step. And we're going to output here a QZB file. This is something that we can actually view or visualize using Chime 2 view. So let me see if I can get that to work. So this is where you would provide a link to that file or drag and drop the file. This is actually, you're not constrained to using a web browser for this. There's actually a tool within Chime that you can visualize from your machine too. You don't have to go to a web browser to do this. But this is one convenient way to do it. So here you see there's just some summary statistics regarding the demultiplex data. There is a visual representation of number of samples with each amount of sequences here. And then you can see each sequence listed with the sequence count in descending order at the bottom. And this is kind of neat. So there's an interactive quality plot with this output. So this is very similar to what you'd see from like FastQC. It's just another way to look at it. So these are, each base has some summary quality data associated with it. So we typically see this drop off as we get to the end of our sequence. So you can use this to kind of determine where you would trim your reads, for instance, for quality. If you need more resolution, you can just drag and zoom in. You can mouse over these different base positions to get summary statistical data for each position. as needed. And then if you need to zoom out, you just double click here. It's kind of difficult without a mouse. Okay. So that's our demultiplex data. The next step is usually to denoise and quality filter. So they were really advocating using data 2. It's one of the newer methods for denoising and quality filtering. It has some built-in knowledge about the error profile for Illumina sequencing technologies. And it also performs de novo chimera checking. So it's I think, one of the preferred methods now for doing this step. So within Chime, there's a plugin for data 2. We're calling the action denoise single. This is for single-ended reads. If you have paired-end reads, then you can use denoise paired, and then this step will also actually join your paired-end reads as well. And I'm not showing every single flag that's available here, but you can certainly add additional flags to have better control over the process. Here we're specifying our input file, which is demux.qza, and if we want to trim on the left, or if we want to truncate by length. And then we're specifying our output files for representative sequences for our OTU table. So I should mention here, in Chime 2, they're no longer calling it an OTU table. So they've changed their language to call it a feature table. And there's some important reasons for that, which I'll talk about on a later slide. But we're going to refer to OTUs as features and the OTU table as feature table. So we haven't yet used our metadata, but in this next step, we can add the metadata to our feature table. So we'll call Chime and then the feature table plugin and the summarize action. So we'll input our table from the last step. It's a QZA file. We'll output a QZV file that contains our metadata. And of course, we have to specify the path to our metadata file also. So now we can visualize our data with the metadata as well. And again, you get this kind of summary data on the first tab here. They're always look for a couple of tabs up here. You get this kind of summary data and some summary visuals as well. What's really interesting about this one, I guess it's kind of trivial, but I find it to be pretty useful. We can look at the data like this. So let's see. What we have here is we're looking at it by barcode sequence, but it makes more sense to look at it by to collapse it by some metadata category. So we can collapse by body site. And now what we have is the number of samples from the gut body site, the number of samples from the left palm, et cetera. And this allows you to play with sampling depth and see what the effect is on the number of samples that you can use and where they're coming from. So at 0, we're suggesting that there's no rarefaction, because everyone's familiar with rarefaction, right? At some point, we need to rarefy so we can do our diversity analysis. So this allows you to select different levels of rarefaction. And as you increase the sampling depth beyond the number of samples that occur, the number of sequences that occur in any samples, they drop off, right? So we've lost one sample here. And as we go up, we can see that we're losing a lot of samples from the right palm. As we keep going up, we're eventually going to lose, all but one of the samples.
Would you explain for those of us?
Yeah, so it's basically when we sequence, we can get a different number of sequences for each of our samples, right? It can vary pretty widely. And so when we're doing our diversity analysis, we need to compare the samples on an even basis. So we would typically randomly subsample from each of those samples so that we have an even sampling depth for all of our samples. And that's the depth that we're specifying here. So this is an interesting way. I mean, you could do this in other ways, but this is an interesting and convenient tool to see where you're losing samples and to help you decide, your cutoff value. Yeah. Yeah, I'm only familiar with the diversity plugin. there you can definitely specify the sampling depth. But we can talk more later. You know, I can look into it. That's okay.
About the clusters that they added more options, like more algorithm, and also for remove chimera, like research, Chime.
vSearch is implemented in Chime too.
Right.
Yeah.
How about clusters?
Clustering is typically done through data or deblur in Chime 2. Those are the two main tools that are implemented.
Right.
Oh, really?
Like interface version, like Chime.
Oh, okay.
And have is a huge step forward.
Yeah.
Expected more options for more. But yeah.
Sure. So that's one way to look at the data after this step. And then the last tab here is the feature detail. So this is what I was talking about before. So instead of listing the OTUs, the way that they list the OTUs or features here is by assigning them another unique universal ID. So they can be tracked throughout the analysis uniquely. And then, you know, the frequency and the number of samples that it's observed in. So this isn't meant to be a tutorial, but just kind of showing you some of the different types of output that can be generated. Next, you'll typically generate a phylogenetic tree. Since I work with ITS data mostly, I'm not that familiar with this step, so I've omitted it here. ITS data is non-phylogenetic, so there are both phylogenetic and non-phylogenetic options in Chime 2. Here, because we're working with 16S data in this tutorial, we're taking a phylogenetic approach. The next thing you can do is start to look at some of the diversity metrics. So with Chime 1, there was a core diversity metrics that kind of output a lot of alpha and beta diversity metrics at once. This is similar. So you can call the diversity plugin and specify core metrics phylogenetic or core metrics non-phylogenetic in this step. So you'll input your tree, your feature table. You can specify that depth for rarefaction in this step. Specify the path to your metadata file, and then the output directory for these results. So I'm just going to show you one type of result. This is the PCOA plot for beta diversity. And those of you that have used Chime 1, you're probably familiar with the emperor plot output. This is going to be an emperor plot. So it's very similar to the output that you would have gotten in Chime 1 with a couple of additions. So what's neat here is these are all your points. Is everyone familiar with beta diversity and PCOA? No? Okay, so it's a measure of how similar or different each sample is from one another. So if they plot close together, they're similar. If they plot far apart, they're different. If we see groups here, in some cases we expect a group of samples to be more similar to each other than to another group of samples. And we hope to see that reflected here in these groupings. So one way you can help to visualize that is to color these by a metadata category. So for instance, we can set the color by body site. So now we can see that we definitely have gut samples clustering together and separate from, you know, the palm and tongue samples. This study took samples over time. So if you're interested in, you know, what happened at a specific point in time, you have options to set the shape of these different points based on a separate metadata category. So we can do days since experimental start. And then these are the different values, and this is the shape for each of them. So if we're interested in day 84, we can change those to cones. And now we can see that, you know, within the gut sample, this is where it ended up. This is an improvement because with Chime 1, you weren't able to do this. And so you had to do a little more work to kind of tease out these details. The last thing here that is different from Chime 1 is the ability to make this a two-dimensional plot. So you can switch your axes around, you can invert them, you know, so that you get the best view, but you can also hide one of the axes to make this two-dimensional. I didn't show you that it was three-dimensional first. Oops. So with the three-dimensional plot, you can, of course, you know, move this around and get different angles. If you hide the third axis, now you have a two-dimensional plot, which is a little better for publication in some cases. And then this little bar diagram shows you how much of the variance is explained by your different axes. So you hope to see some kind of break here where, your first couple axes are explaining most of your variants. And this is also new from Chime compared to Chime 1. Okay, just a couple more things. So some tips here that you can add the help flag at anywhere in the command, and it will override all of the other flags. So let's say you have your long command kind of mostly done, but there's something you're uncertain about. You can just tack on the help flag and then read the help output, decide how to modify your command, and then you can just use your up arrow to repopulate your command. So you don't have to start over. You know what I mean by that? If you're using Linux and command line, you have a history of all the commands you've entered. So it's a convenient way to not have to rewrite your whole command to get that help. And then finally, I know this is kind of low on the slide, you can use Chime tools, import and export, to step in and out of this at any point. So you can export your data from the QZA file, which is basically just extracting it and placing that data file separately in some location. Okay, 2 more outputs I think are interesting to look at. One is alpha rarefaction. So this comes back to that rarefying step. I don't know if everyone is familiar with this type of plot, but typically we'll generate this type of plot to kind of get an idea of how good our coverage is. So did we sequence deeply enough? And what we're looking for here is for this plot to plateau. So this is kind of a random subsampling at different sequencing depths and looking at how many OTUs are observed at each depth. So if we're sampling deeper but not getting more OTUs, that means that we've probably covered all the diversity in our sample. So this is similar to the Chime 1 output. You can select different metrics to display here. You can also collapse this by metadata category. So you can collapse this by body site. This is pretty standard. And then down here, this is kind of like what we were looking at before. It's another way to try to pick cutoff values for rarefaction. So you can see the number of samples that were included at different sampling depths. So obviously for this metadata category, we lose a lot of samples if we go above 1,000 for our rarefaction depth. And the last thing that we'll look at is the taxonomic bar plot. This is usually of interest. Okay, so this is a big improvement on Chime 1 output. Chime 1 output was an HTML file, but each taxonomic level was a separate plot, and you'd scroll way down the page to try to look at species, for instance. Here you can select the taxonomic level, so you can go down to species if you'd like. Probably not recommended. You can also stick to like class level. You can change the color scheme here, whereas in Chime 1 you couldn't do that at all. So you can pick between discrete and continuous color schemes. You can also sort, and in Chime 1 you couldn't sort. So here you can sort by metadata category, for instance body site, and then you can actually add secondary sorting factors. For instance, you could sort by the abundance of a certain taxa that you're interested in. And then you'll have these sorted within, so 2 levels of sorting here. So that's a pretty interesting new feature. You can download this as a SVG, which is a vector image, so you can reproduce it at any scale and it will be high quality. And you can download also a CSV file for this output. So while we're here, 2 things we haven't looked at are these 2 tabs up here. They've been present for all the other outputs, yeah? Not right now. No, but that came up during the workshop and they're adding features continuously. So the other thing is that if you're interested in a feature that isn't available, you can go to the forum and there's a section of the forum dedicated to feature requests. So in all the output, we've had this peak, which is not terribly informative, but it does give you the UUID for this particular output, its type and its name. Not too useful. But here's where the good stuff is. So this is the provenance. So you can actually kind of drag these around to get the best view possible. But what we're looking at here is that at each step, we have a node, and the box is the action or execution that was performed at that step, and the circle is the output. So the best information is contained in the box, which tells you, first of all, as I said, all of the executions are also assigned this unique ID. So we can later trace back to it uniquely. It gives you the runtime, a lot of really detailed data that you wouldn't otherwise take notes on, most likely. Most interestingly, every single Python package that you use in this calculation is included here. So there are hundreds of them, and they're all listed with the version as well. So if later on you find out that there was some critical error in some version of some package that you were using, you can go back and look to see if you actually use that package in your analysis, and you can rerun the analysis with an updated version. This is also useful, this whole data provenance thing can be useful for automation as well, and reproducibility of your analysis. So I think this is probably the most powerful addition to Chime 2, in my opinion. So just coming back to the OTU versus feature table, one of the reasons they did this is that with Data 2, it's becoming more accepted to cluster at higher sequence similarities. So you can go to 99 or even 100%. And now we wouldn't call these hypothetical species, but subspecies, sub-OTUs. And this is due to just the quality that you can get through the Data 2 algorithm. So by calling these feature tables, then we can have, rather than OTUs, we can have sequence variants, which are the clusters at higher similarities. And you can later collapse the feature table to different taxonomic levels. So you get your sequence variants, you assign your taxonomy, and then you can collapse to genus level if you'd like, or class, or some other level. Yeah. That's the terminology they were using. I think that's the trend. It's kind of semantics, as long as you're aware of what you're doing. But yeah, that's the terminology they were favoring. Yeah. And then the last implication of this is that by moving to this new terminology and kind of generalizing things to feature tables instead of OTU tables, we can have features that aren't OTUs. We can have features that are KEGG category counts, for instance. So it's one way to kind of open up the platform to other types of analysis like metagenomics or metatranscriptomics. I mean, there's more that would need to be done for this to be usable for those approaches, but they definitely have that in mind as they were rewriting this code, trying to broaden the scope of Chime 2.
Would you just share
Yeah, so KEGG categories have to do with functional genes. So if you're using something like PyCrust, you might be familiar with this. But we can, that's actually pretty complicated. I don't know if I can do a good job of explaining it.
I think that's enough.
Yeah.
Some people are really entry level, and so they at least get an idea, okay, that relates to.
Yeah. So this is getting toward the functional stuff rather than just, taxonomic stuff. And the experimental plugins I wanted to mention, I know we have someone who does a lot of data science, but I don't see him here today. I don't know his name. What is his name?
Yes.
Yes. Yeah, so he gave a nice talk on sample classifiers actually a couple weeks ago, impromptu on the whiteboard. I found it very interesting. But there is a sample classifier that is in early stages of development. It's considered experimental. You can Google, I had a hard time navigating to this through the webpage, but if you Google Q2 sample classifier, they do have kind of a tutorial on that. And that's for using supervised learning methods to predict sample metadata values. So you'd have some set of samples. You'd see how the samples fit into different metadata categories based on the taxonomic profile. And then if you have a new sample, you could theoretically figure out which metadata category it belongs to based on its characteristics. You're trying to classify new samples. And then longitudinal is to look at studies over time or over some environmental gradient, which I don't think was implemented in Chime 1. So this is also experimental. These were included in the workshop, but really cursory, so I can't go into much more detail. But if you Google those plugin names, you'll find more information about them. Yeah.
When you say experimental plugins, what is that?
They're not fully functional yet.
Okay.
Yeah. They're supported, they're developing them actively. Yep. So it's a good place, if you're interested in those, it's a good place to get into the discussion and say, I'd like to see this feature in this plugin. So here are some resources. These are pretty easy to write down. So Chime forum, you'll find, like I said, feature requests. You can indicate any bugs that you find, and you can get general support with any questions that you have with your analysis. Tutorials and documentations are pretty expansive on the Chime website, so docs.chime2.org. And you'll see when you type this in, you'll get a / version number. So they're updating the version of Chime 2 like once a month. So you want to make sure that you have the newest version and that you're looking at the docs for the correct version. So what they recommended was just using Miniconda, you can have a new environment for each version that comes out and just have multiple versions installed. But try to use the newest one because that's going to be the most functional. If you want a general overview, the moving pictures tutorial is really good. That's the one they selected for the workshop to kind of go through with us. But it uses single-end reads. So if you're using paired-end reads, they suggested this Atacama tutorial, which I haven't personally gone through myself. And if you're using already demultiplexed data, then importing data tutorial will show you how to squeeze that into Chime. So if you use the facility on campus, they typically provide demultiplexed data, and so that would be important for you. And finally, Greg Capareso is working on this open source applied bioinformatics book, which I think is going to be focused on amplicon, marker gene surveys. So I think it's worth checking out if that's the type of work that you do. Readiab.org. It's available statically and then also as a Jupyter notebook, so you can go through and I think work through the examples right in the text. And that's all I have. Thank you. So I can try to answer questions, or we can talk a little bit about Chime and Mother and what people are using and what they like.
Michael, I'm sorry, I missed the first part of it. So can you explain where you got this information, workshop that you attended and what it was like?
Yeah, so they're doing a series of workshops, the developers. This one was at, so I should say the Microbiome Center supported my attendance at this workshop based on the poster contest last year, which I was grateful for. But it was down at the National Institutes of Health campus in Bethesda. And there were about 100 people there. So it was a large workshop. And the developer of this software and two of his main programming guys were there giving the workshop. And so that's where most of this information came from, was from their slides. It was a 2-day workshop, so it's not all here, but it was pretty informative. A lot of it was going through those tutorials that are available online. So they also rely on those as like a primary source of instructive information.
So just to follow up on that, what was the cost of the workshop, and would it be appropriate for someone who is complete novice for this sort of work?
The cost was about $400 for graduate student registration. I'm not sure. I can't remember the costs for others. I think if you just took a few minutes to look at the website, basics of the tutorials, and if you had an idea of what a marker gene survey consisted of and what the goals are for a survey like that it would be appropriate for a beginner. Yeah, they were all different levels of experience there. And they took a lot of time. If we needed to ask questions, they would stop, and the questions were all different levels. So how many people use Mother? Does anybody have a strong opinion? No?
I like the developers more.
Yeah.
On the forums, and you ask questions on Mother, they assume that you know a lot, and they aren't very helpful in our knowledge, whereas it's very different. There's a whole range of people who look at those boards and look at the questions. And they don't seem to mind, oh, I didn't know. I don't really see that very much when I'm on the Mother board. So that's kind of like the Shreves we use Chime.
Of course, you should always search the forum first for your question.
Yeah.
I have knowledge that we can talk about that. And more in a biological way, how to apply bioinformatics tools and drag more life on it.
Yeah, I think that's good.
That was a particular reason.
Yeah. Mara?
Like you said, yeah, they are more like you should do your survey or search of something that online by yourself. But they provide a lot of online. So now will be a huge step forward. They provide tutorials on YouTube now.
Oh,
They will have random interface, like features to visualization. I think more than
you can sway. Okay. Yeah, they really seem very interested in bringing a lot of people on board, making it accessible. And definitely, if you have feature requests, now is the time to mention them.
I don't know if they got into this at all at the workshop, but aside from saying that they're switching from 97% clustering to the 99 to 100%, I thought my understanding, and this was, I don't know, probably like second or third hand information, was that they were switching the way they did the clustering completely for Chime 2. Is that true? Is that what using data is for?
Yeah, I believe that by using Data2, you're able to cluster at these higher similarity values because of the improved algorithms for quality filtering and fully de novo primary checking. Where in Chime 1, it was an open reference.
Well, and they were using like research and clustering
Yeah, yeah.
for Chime 1, so that's the switch there.
Yeah, I might have just misspoke, but yeah, in general, Data2 allows for the higher sequence similarity. But it works, I think it works best with Illumina data. I think that's Is that the standard? They stayed away from indicating standards, I think. Yeah. So you get to struggle with that and decide on that. Right, but so what you're doing is you're clustering at a really high level, but then you're assigning taxonomy also. So now you're going to have, all your taxonomic ranks and later collapse back down to your genus level. Right.
You would get much additional phylogenetic signal from 99% clustering. There's not enough information for a lot of those. So
Right. Yeah.
when you collapse, some information. But I think the bigger difference from my understanding, which is like I said, is that they were completely switching the way they did that cluster. So the way they did it before wasn't. It was resulting in a lot of noise.
Yeah, spurious. Yeah, this is improved cluster. I don't know if Data2 is implemented in Chime 1 or not. I never tried to. No. Yeah. So these are just Data2 and Blur in Chime 2.
Could we go back to your diagram of the different steps? One of the things that I was thinking as I was looking at that, and that was really nicely done, is that there are opportunities for anyone who is working and researching any one of those places. Like if you've done a lot of sample prep, for example, and you have an understanding of kind of the ins and outs of sample prep, that would be a really great presentation for next semester. And so if you're looking to go to TV or practice presenting, or you want to be able to be more fluid with methods that you're using that you're going to have to present for your comps or something like that, I would suggest that you get in touch with either me or Tim Yushiro to say, yeah, I really want to talk about library generation. And it would be great, because I think there are a number of people, there are people in the Microbiome Center who are well beyond that, but there are people who are really just sticking their toes in the water, and so they would.
Yeah, and there's a lot of
stuff there.
Yeah, like especially right here. There are a lot of ways to do this.
Yeah. And what is it that you like? Like each of us, tends to have a thing that we become specialists in. We sat down and we did all the backgrounds of find every little nook and cranny. But we don't do that forever, right? We get into something and then we do it, and then we know every hair in the belly button, right?
Yeah.
So please, you know, if you have something like that that you can easily. be great. Well, thank you so much. This was really useful.
You're welcome. Thank you.